Great, can you guys see that?
Yes, it's perfect.
Excellent. Okay, so hi everyone. I want to first thank Wake Up Narcolepsy for the opportunity to speak today. I'm really excited to be here and to share my data with you. My name is Sara Pintwala, and I'm a PhD candidate in Dr. John Peever's lab at the University of Toronto. Like John, I have 2 animals running around my house, and so if you hear any noises from them, I apologize in advance. I am going to be giving you a talk today on my PhD project called Transplanting Orexin cells in Narcolepsy. When we wake up in the morning after a good night's sleep, we feel refreshed. That's because sleep is a behavior characterized by rest and restoration of vital processes. I bet you all look like this when you wake up in the morning. I know that I sure do. Our understanding of how cells in the brain control sleep and wakefulness have come a long way in recent years. And one system in particular, the orexin system, is of paramount importance to wakefulness. Orexin is a neurotransmitter responsible for producing and maintaining wakefulness. This chemical ensures that when we are awake, we stay awake for periods of time. Orexin is produced in an area of the brain called the lateral hypothalamus, which is outlined here with this square on a side view of the rodent brain. Within this specific region of the brain resides orexin neurons, which are pictured here on the right with red fluorescence. There is no other region in the brain that produces orexin. These cells are only located in the lateral hypothalamus. And when these cells are lost in narcolepsy, there is no other source of orexin in the brain. In this slide, I'm showing you magnified images of the lateral hypothalamus. On the left is an individual who does not have narcolepsy, and you can see the presence of orexin indicated with black coloring. On the right is what the brain looks like in an individual with narcolepsy. You can appreciate that in this individual, that orexin is absent. When orexin neurons are lost in narcolepsy, it makes a disconnect in how the brain typically produces and maintains wakefulness. As a result of this, individuals experience symptoms relating to sleep and wake instability. Here listed are the symptoms of narcolepsy. This includes excessive daytime sleepiness, sleep disruption, sleep paralysis, hypnagogic hallucinations, and cataplexy. I'm sure that you're aware of these symptoms as some of you experience them yourself. Therefore, I won't go over the details of all of these symptoms of narcolepsy, but I would like to focus on cataplexy. When individuals experience cataplexy, they have a sudden and involuntary loss of muscle control during wakefulness. Although the subject becomes paralyzed, they are conscious and fully aware of their surroundings. Cataplexy is a behavior that we're really interested in the Peever lab and is a major focus of our research. As sleep researchers, we're really lucky because we in the laboratory have a mouse model of narcolepsy. By deleting the gene which encodes for orexin, we're able to make a mouse who does not express orexin at all. This is the orexin knockout mouse, and it recapitulates the symptoms of narcolepsy, including cataplexy. I have a video here of cataplexy in a narcoleptic mouse. And what you can see is that this subject is awake, walking around his cage, when he begins to experience cataplexy and collapses to the floor. His cage partner actually whips him with his tail accidentally while running on his wheel, and you can see how the subject is unable to respond to his environment. Using animal models like the orexin knockout mouse, we're able to investigate narcolepsy in the laboratory, including testing for new therapies. From a clinical perspective, managing the symptoms of narcolepsy focuses on behavioral and pharmacological-based interventions, but there is no permanent cure for this disorder currently. Behavioral and pharmacological strategies ameliorate some symptoms of narcolepsy, but have limitations for the 3 million people worldwide who have this disorder. As such, additional strategies are needed for the treatment of narcolepsy. The overarching aim of my research is to find a new treatment for narcolepsy by transplanting cells in the brain. In mice who have orexin, shown here at the top with the red cells, we see normal sleep and normal wake behavior. When the orexin neurons are lost in narcolepsy, shown here, we see excessive sleep and cataplexy. The premise of my PhD project is to grow orexin cells in a dish and transplant them into the brains of a mouse model of narcolepsy, shown here on the bottom. Because cataplexy is a major focus of research in the Peaver lab, we are interested in studying this symptom of narcolepsy specifically. Overall, for the field of cell transplantation, the objective is to alleviate symptoms and treat disease. As such, the primary objective of my project is to determine if transplantation of orexin cells into the brain will alleviate cataplexy as a novel therapeutic strategy for this disorder. For several reasons, narcolepsy is a suitable disorder to test new treatments like cell transplantation. First, it is a single type of cells which are lost, the orexin neurons of the lateral hypothalamus. Moreover, there are a number of robust and well-characterized rodent models currently available for narcolepsy, with etiology almost identical to the human disorder. The final condition of testing cell transplantation is a question of resources. If we're going to transplant cells in the brain, we need access to cells identical to the ones lost in disease. For this, we now have a novel orexin cell line supplied to the Peaver lab by our collaborators in the Belsham lab at the University of Toronto. Overall, this means testing novel treatments for narcolepsy, such as cell transplantation, is possible. The cells I'm working with are called M-hypo-A ORX GFP4 cells. This is a mouthful, so for my presentation, I will simply call them the orexin cells. They are an immortal cell line, meaning that they have been transfected with factors enabling them to divide in the dish over time. What I'm able to do with these cells is grow these cells in a dish and then transplant them into the brain. On the right, or on the left rather, I'm showing you an image of live cells growing in the Peaver lab in the dish. A major advantage to using these cells in a dish is that over time, I can grow billions of orexin cells for transplantation. Using cell transplants to treat neurological disorders is common in other circumstances, but not for narcolepsy. Experiments in Parkinson's disease dating back to the 1970s pioneered the field. In the same way that people with narcolepsy lose orexin neurons, people with Parkinson's lose dopamine neurons. Dopamine is a chemical messenger like orexin, but functions to control movement. When dopamine neurons are lost in Parkinson, there is a decreased level of dopamine in the brain, which is shown to you here in Figure A. When dopamine neurons were grafted into human patients with Parkinson's, an increase in dopamine in the brain was observed, shown here in Figure B by the darker red and yellow coloring. These experiments have been reproduced many times, showing success in reducing symptoms and tolerability over time. Since then, cell transplants have been explored for the treatment of a variety of disorders, including stroke, epilepsy, and even schizophrenia. The idea is to now apply this technique as a therapeutic strategy for narcolepsy. The cells I work with were made by our collaborators in the Belsham lab in 2012, but nobody in the Belsham lab to work with them. We like to joke that these cells are like the forgotten child of the Belsham lab because nobody worked with them. To me, however, this was very exciting because I became the first person ever to work with these cells, but it also meant that I had to validate their character. For the first 2 years of my PhD, I worked with my cells in the dish, also known as in vitro. And specifically, what I had to do was show that these cultured cells, 1, express orexin, and 2, secrete orexin. Because the premise of cell transplantation is to replace lost cells, I had to show that these cells indeed express orexin. But expressing a chemical messenger is far from being able to actively release it. There are many complicated processes that have to occur within cells to get the orexin within the neuron into the external environment. And if we're going to replace orexin neurons in subjects with narcolepsy, we need to make sure that our cells can functionally release this chemical messenger. To detect orexin expression in cells, I performed a technique called immunocytochemistry. This is a technique commonly used in science to identify proteins in tissue using color. In these experiments, we culture cells, shown here in step 1. After this, we apply antibodies that bind specifically to orexin neurons, shown here in step 2 by this black protein. After this, we apply an antibody which binds specifically to the orexin antibody that emits a color, shown here in red. Using microscopy, we can observe cells for the visualization of orexin, shown here in figure 3 and figure 4 with red fluorescence. To detect and quantify orexin released by cultured cells, I performed an experiment on live cells. Neurotransmitters like orexin are transient in the brain. Orexin neurons in the brain make this chemical and then store it in their cell bodies. It's only when stimulated that they actively secrete orexin into the fluid which bathes the brain. What I did was grow cells in a dish, shown here in step 1, and I treated my cells, here in step 2. Orexin cells in the brain respond to changes in glucose level, and so I manipulated glucose levels in the dish. After this, I collected media and I quantified the media using a technique called enzyme immunoassay. The details of enzyme immunoassay I won't go over here, but it's a technique that allows us to identify and quantify protein levels present in solution. In this slide, I'm showing you the results of these experiments. In figure A, we see many cells expressing orexin, indicated by red fluorescence, with a magnified image here showing individual cells in figure B. And so I was able to answer an essential question to my project and validate that this novel cell line indeed expresses orexin. I also found that these cultured cells functionally secrete orexin. I was able to detect media in the high glucose condition, and I found that there was far more orexin released by cells when they were probed with low glucose solution. The importance of this to my experiment was twofold. I was able to show that cultured cells express orexin, and 2, that these cultured cells are capable of secreting orexin in a similar manner to orexin neurons present in the brain. The next set of experiments had the overarching goal of finding a way to control the activity of cultured cells. We wanted to find a way of controlling the activity and orexin released by cultured cells specifically. This would confer to us a major advantage as it would allow us to manipulate orexin cells in the brain after transplantation. The technique we chose is called chemogenetics and involves driving the expression of synthetic receptors in cells. The synthetic receptor I chose is called H3DQ, which is a mouthful, so I will refer to it simply as a synthetic receptor. Chemogenetics as a technique is quite complicated, as I'm sure you can appreciate from this slide, but overall it is used in neuroscience to activate cells in the brain. When a drug called CNO binds specifically to the synthetic receptor, it turns on and activates neurons. This drug called CNO has been designed in such a way that it won't have effect anywhere else in the brain, only on neurons that express this receptor. By administering CNO, we can therefore turn cells on when we want to for as long as we want to. And as an extension of this, we can program orexin cells to release orexin on command with the use of this system. In this experiment, I again used immunocytochemistry to identify orexin cells growing in the dish. In these images, the activity level of cells is indicated with dark staining. So the darker the cell, the more active it is. When cells expressing the synthetic receptor were treated with CNO, shown here in figure D, the cells were very dark. And you can appreciate how much darker cells were in this group when compared to the other 3 groups of controls. Overall, we found that orexin cells expressing the synthetic receptor were far more active when treated with CNO compared to other groups shown here in figure E with the red bar on the far right. I next performed a secretion assay to determine if we can boost orexin release using this system. A cell becoming active is far from being able to release a chemical messenger like orexin. There are numerous molecular processes that have to occur to get the orexin within neurons into the external environment. And so, although my cells became more active with CNO, we had to look at orexin release specifically. When orexin cells expressing the synthetic receptor were treated with CNO, we found a great increase in orexin release, shown here with this red bar. In sum, these experiments indicate that we can use synthetic receptors to increase the activity and orexin release by cultured cells. In the next set of experiments, we performed behavior to determine the effect of cell transplantation in a mouse model of narcolepsy. My goal was to determine if transplanting cultured orexin cells reduces cataplexy specifically. Orexin cells were grown in the dish and transplanted to the brain of a mouse model of narcolepsy, the orexin knockout mouse. To do this, we perform brain surgery on mice who are anesthetized so they don't feel any pain at all. During this surgery, we can target cells to a very specific area of the brain, which is shown here on the right. The area we chose as a surrogate for the orexin cells was the dorsal raphe, outlined here with this blue circle. The dorsal raphe is a unique and specialized region of the brain of importance in narcolepsy. In these mice, we observed cataplexy to determine the outcome on behavior. When orexin cells were transplanted into the dorsal raphe, I found a reduction in the number of cataplexy episodes when compared to controls shown here in figure A. The reduction in cataplexy was substantial, as subject had a 70% reduction in cataplexy episodes. Subjects also had a reduction in the amount of cataplexy, meaning they spent less time in cataplexy over the day, shown here in figure B. I found there was no change in the duration of cataplexy episodes between groups, meaning that the duration of individual cataplexy episodes were unchanged by the intervention, shown here. Overall, this data indicates that transplanting orexin cells reduces the severity of cataplexy, but does not abolish it entirely. I then performed immunostaining for orexin in transplant recipients to determine how many orexin cells were present post-transplant. In figure A, I'm showing you a picture of the mouse brain from the front, and I found that there were orexin cells present in transplant recipients, shown here with red fluorescence. In figure B, there's a magnified image of the transplanted cells where you can see many orexin cells present in the grafted site. In figure C, at 100 times magnification, you can see the integration of an individual cell, where numerous extensions were made into the host tissue. When quantified, we found approximately 250 cells present. I want to take a moment to focus on this data because it is significant to my project as well as to the field. With my intervention, I took mice which are devoid of orexin, and by transplantation, made in them orexin expressing cells exist. I next performed a series of experiments to serve as a positive control for my project. In these experiments, we wanted to further explore orexin replacement in narcolepsy by injecting orexin proteins directly into the brain. What we really wanted to know from these experiments is, can we change cataplexy by injecting orexin to the brain in the same way we did with cell transplants? To do this, we took purified orexin and suspended it in the fluid which bathes the brain called CSF. We then performed surgery on a mouse model of narcolepsy, similar to our transplant work, and installed a guide cannula in their brain that enables us to make precise injections into the brains of living animals. We also implanted electrodes in these animals for electroencephalographic and electromyographic recordings to identify cataplexy, similar to the polysomnographs that some of you undergo. After this, we made careful injections of orexins into the brains of mice and observed for cataplexy. We found that injections of orexin directly into the brains of orexin knockout mouse decreased the number of cataplexy episodes and the amount of cataplexy, shown here in figure A and B. There was no significant change in cataplexy episode duration across all 3 groups, shown here in figure C. This data is highly significant to my transplant work as it is consistent with my primary findings. In narcoleptic mice, we may use orexin replacement by cell transplantation or direct injections to modify their behavior and make them exhibit less cataplexy, but we were not able to abolish cataplexy entirely. In summary, I have shown that cultured cells both express and release orexin according to normal bodily cues. Orexin cells are candidates for control by chemogenetics in the dish, exhibiting an increase in activity and orexin release. Transplantation of orexin cells to the dorsal raphe reduces cataplexy in a mouse model of narcolepsy. Injecting orexin peptides to the brain in a mouse model of narcolepsy reduces cataplexy akin to that of cell transplantation. What I'm now working on in the Peaver lab is merging my in vitro chemogenetics work with my behavior work to determine if the activation of transplanted orexin cells will further reduce cataplexy. To end my presentation, I would like to relate my findings to human health. This is, after all, the goal of scientific research. My PhD thesis is a proof of principle project. I'm trying to provide evidence towards using cell transplants to treat narcolepsy. If I'm able to transplant cells into the brains of a mouse model of narcolepsy and rescue this disorder in animals, then this could be a way to treat narcolepsy one day in humans. With that, we are at the end of my presentation. I would like to thank Wake Up Narcolepsy for their support of my project and for the opportunity to speak today. I would also like to thank the Canadian Institutes of Health Research for funding our research, as well as the Peaver and Belsham Labs for their support in my project. Thank you for your time, and I'm now happy to take any questions.
Thank you, Sarah. That was extremely informative. We do have some questions. So I'm going to read one by Shannon Hall for you. As it is understood that people with narcolepsy are likely to be overweight, even if they consume less calories than non-PWN, my understanding is this, in connection with loss of orexin, I think that this is in connection with a loss of orexin. If your research comes to therapeutic use of the orexin, is it likely that this would also be affected as well as the level of cataplexy and, of course, the sleep schedule?
So I do think that it does have relevance to the metabolic features of narcolepsy, but something that we really have to discuss with the context of cell transplants is that their orexin release is very local. So I didn't show you guys this, but I actually tried transplanting orexin cells to the lateral hypothalamus, and what I found was that there was no effect on cataplexy in these subjects. That's really when we went back to the drawing board and decided to consider the dorsal raphe as a surrogate region for these cells because this specifically is important for cataplexy. I do think that it is possible for us to extrapolate our results to help reduce the other symptoms of narcolepsy, but that really depends on us understanding how those metabolic features come about in the brain. And so it would be possible to transplant cells to other regions to ameliorate those symptoms, but because we don't understand it fully, it's not possible at this time.
Thank you. In the other diseases that you mentioned earlier in your talk, you referred to Parkinson's and strokes. Are there treatments already being utilized in terms of cell replacements for those disorders with a view to that being applicable to narcolepsy? And I mean in humans.
Parkinson's is really the classic disorder for cell transplantation, and there have been a number of clinical trials which have come far from these experiments. They mostly use cells that are stem cells in Parkinson's transplantation research, but overall they have success in reducing symptoms and they have tolerability as well in subjects. In order for us to really extrapolate this research into humans, we would need human orexin cells. Generally with cell transplants, it's a no-no for you to cross species. And what I mean by this is to take a mouse orexin cell and transplant it into a human. This produces something called graft versus host disease and results in immunological problems and leads to the death of cells. So really, to extrapolate our research to humans, we would need orexin cells isolated from humans. And this is something that my collaborators in the Belsham lab are actually working on. Dr. Belsham, throughout her career, has made over 200 cell lines, and she's currently working on trying to isolate orexin cells from human patients so we can then bring this project forward and have it have clinical relevance specifically.
Thank you. So with regard to the cells that you had generated, in terms of their secretion, what is their lifespan?
So these cells are an immortal cell line, which means that they divide in the dish indefinitely over time. They've been transfected with a number of factors that enable them to do this, and so in a way they are immortal. A big advantage of this is that we can culture many cells over time in the dish, but as soon as these cells come into contact with another cell or tissue, they stop dividing. And so that's why they continue to grow and are immortal in the dish, but when we transplant them in the brain, they become static.
That sounds like a huge problem.
It could be, but it has been well validated that these cells do stop dividing once they come into contact with other cells or tissue. It's been validated by our lab, some of my other work, as well as other people who use immortal cell lines, and it is a common and consistent feature of them.
Okay, thank you. I have a question from Arnold Gendelman, who asks, what did you use for the sham transplant?
For the sham transplant, the procedure was the same as transplanting orexin neurons, but instead we made a fluid injection to the dorsal raphe. So instead of there being cells present, it was the vehicle that the cells were suspended in for transplantation.
Thank you. The next question is based on the previous response. Would you, I think the one before this last one, would the cells be able to be extracted from people in the same sense that people donate organs?
Yes, exactly. And this is exactly how Dr. Belsham received these tissues. She had brain tissue isolated from 2 individuals who were organ donors. And with the help of a neurosurgeon at Sunnybrook, she was able to extract neural tissue from these 2 human subjects for the generation of immortal cell lines.
Thank you. So you are listed as a PhD candidate. This sounds like a really polished thesis.
Thank you.
Are you coming to the end of that? Will you be staying with Dr. Peever?
So I am almost at the end of my PhD. I only have a couple more experiments to run, which I'm performing now in the lab, and I have most of my thesis written as well. So although I've been so happy to be with John for my PhD as well as undergrad, I was in his lab. I will be accepting a postdoc position with Dr. Fuller at the University of California, Davis campus. So I will still be in the field of sleep research, but I will be moving on to a new lab.
Congratulations.
Thank
Can
you.
I ask what precipitated your interest in narcolepsy?
I really think for a very long time, I was really interested in animal behavior. I started doing research during undergrad in year 2, where I stayed throughout year 3 as well. And this was an animal behavior lab where we were studying the effect of pain and anxiety on memory. And I took a sleep course in 3rd year, and I just became honestly really obsessed with this. After that, I applied to Dr. Peever's lab, where I performed my 4th year undergraduate thesis. And it was really through his research interests and his mentorship where I became really invested in this disorder.
Thank you so much for your contribution to the narcolepsy community, Sarah. Are there any other questions? I think we are good on those. Thank you again, and all the best with your Viva and your ongoing work.